Okay, great. So good afternoon. Today's topic is imputation, and the motivation for this, I worked on imputation and diploids as a postdoc over 10 years ago and have dabbled in it off and on for polyploids for a number of years, and just have gotten a lot of questions recently about what are best practices and realized I didn't think there was a good reference on this. So this is my attempt to synthesize some of what I've learned over the last decade or more. And we can kind of think of two different scenarios here, broadly speaking. So scenario one, which is the one I'm going to focus on today, is a situation where you can't make any explicit assumptions about the mating design or the population structure. So you don't have that as an advantage to exploit. And that's a common situation when you're looking at a GWAS for association studies and many genomic selection applications. Scenario two is very much in line with what we've been hearing about already today, where you have a known population mating design, and you can use the same methods that are used to do the linkage mapping, essentially also impute missing data. So if you're in a situation like scenario two, figure out how to do that in the context of the software that's already available to you. And I'm going to talk about today is this scenario one. And most I'm going to focus on today is marker data sets that are generated by sequencing technologies, whether it could be whole genome sequencing, restriction enzyme-based complexity reduction, amplicon sequencing, so on and so forth. But the same methods and software can be easily applied to SNP arrays. So I'll touch a little on that at the end. Okay, so for panel imputation, again, diploids, this has been worked on for a very long time, and I would say that the kind of gold standard represents a haplotype-based approach, which is using a hidden Markov model like we've heard about, but instead of it doing it instead of being able to exploit, let's say, something very clear about the transmission genetics, it has other, it has to make assumptions about like the number of unique haplotypes in your panel and so on. But there's still software that does this pretty well. Beagle being one of the most famous ones. More recently in plants, people are using this thing called the practical haplotype graph. But in any case, for polyploids, we're not at that level just yet, as far as I know. Some people have made claims that they have software that does this, and I've tried some of it out and have had pretty mixed results. I'm excited to hear a bit more about the polyhaplotyper software tomorrow, because it seems from the abstract that maybe it's one of the first software that is doing this in a meaningful way for polyploids, but we'll find out more. So anyways, in the meantime, what I can tell you about are things that we've tried that don't really exploit a haplotype approach in the genetic sense of it. And the two methods I'll talk about are the first one being this approximation called the multivariate normal EM algorithm. And the second one is a classic machine learning algorithm called the random forest. Before I do that, I wanted to talk a little bit about the variant call format, or VCF, which is, after trying a number of different things in my career, I've just decided to try and write all my new software to work with VCF, because it gets to be a pain to convert back and forth. And I think for many of you, the data sets you're getting from your service providers are in many cases in this format. So I just wanted to take a moment to just highlight some of the features of VCF that will be helpful to understand the software I'm going to tell you about. So the beginning of every VCF file has what's called the metadata section, and it all begins, all those lines begin with two hashtags. Then you have a single line that has the header, which starts with a single hashtag, and it has a set of fixed fields that must be present in this order. There are the chromosome, the position on the chromosome, an identifier for that variant if there is one, the reference and the alternate alleles, a quality score for that variant, and then whether you've applied some filter. And if you don't have, let's say in this case, this data set doesn't have a quality score or there's no filtering applied, you can see that period is used. Period is the symbol for missing data in the VCF. The last column, info, has a number of statistics about that variant. Common information is allele frequency, the depth, the number of samples, and I'll talk a bit more about that later on. And then you have a column called format, and then after that you have the data for each of the individuals in your population. So this example here has a single column for the first individual, and you could have many more after that. Let's look at the structure of the format and one of these individuals here to get a little bit deeper dive. So format is a colon delimited list of what are called genotype keys, or just codes that describe, again, various properties of that particular individual at that particular variant. And here it's one of the most common keys that you find in VCF files. AD stands for allele depth. It's a comma delimited list of the number of reads counted for each allele. So my examples here are all going to be bi-allelic, 2 alleles. So this example, there's 1 read of the reference allele and 7 reads of the alternate allele for that particular sample. That's what 1,7 means. You add those numbers up and you get the total depth for that sample. So that's what the DP stands for, total depth. GT stands for genotype. It is what you could call a slash delimited, because a slash is separating each of the predicted states of the chromosomes at that particular locus. So this is a tetraploid example, and a 0 stands for the reference allele, the 1 stands for the first alternate allele. If you have more than 1 alternate allele, it'd be 2, and so on. So this particular sample is predicted to have 3 copies of the alternate allele and 1 copy of the reference allele at that position. GQ stands for genotype quality. That's a Fred scaled error, measure of the error of the predicted genotype. So in this case, that's a value of 5. Let me say a bit more about what does that mean? What's Fred scaled error? So every predicted genotype has some probability associated with it. And conversely, there's an uncertainty, which is 1 minus that probability of the prediction. And Fred is essentially a logarithmic scale. So the higher the quality score, the smaller the uncertainty is growing exponentially logarithmically for that. So it's named after this program Fred from the Human Genome Project a very long time ago. But to give you some examples, if you have, let's say, a 10% uncertainty or a Q value of an error of 0.1, that's a GQ score of 10. By the time you go down to a Q score of 0.01, so 1% error, that's a GQ of 20, and so on. So for example, 5 means GQ of 5 in this earlier example. That means over 10% error associated with that prediction, which is a very uncertain prediction based on only 8 reads in a tetraploid sample. Okay, so I'm going to try and talk a little bit about now the underlying mathematics or probability model associated with how these different genotypes and their characteristics are predicted. And the basic mathematical model is called the binomial model, which is the mathematical term for flipping a coin a number of times. Okay, you flip a coin, you can get heads or tails. You flip a coin a bunch of times. That's the same thing like observing a number of reads for a particular sample at this particular locus. So the total, let's say, depth of the alternate allele is this random variable. We have the n is the total number of reads. Here's the comma delimited AD field, which has n minus k for the reference allele, k for the alternate allele. And then you have this probability of observing the alternate allele, which like in a fair coin is a half. You get equal probability of heads and tails. And if you had 0 error or bias or other complexities, you'd expect that probability to equal the dosage of the alternate allele divided by the ploidy. So like, for example, in a tetraploid, those p-values could be 0 to 1 in increments of a quarter based on, again, that dosage. In real data sets, that's not what happens. You have to account for these additional complexities like sequencing error, allelic bias, and furthermore, even with those, you just see more variation than you would expect mathematically. That's called overdispersion. And many of you already know about this nice package, UpDog, and we're glad to have David here in the audience who helped to develop that, who was the principal developer of that. And I'm going to be using that software in what I'm going to tell you about for most of today. So this package UpDog has a lot more information about these kinds of models. Just to give you a visual of what's going on here, this is showing you the distributions associated with a tetraploid example where you have the true dosage is either 1 or 2 copies of the alternate allele. On the left, you have 20 total reads. In the right, you have 40 total reads. And you can see at a lower depth, the black and the red, there's more overlap in like between, so with 20 reads, if you're between 5 and 10 reads of the alternate allele, there's some uncertainty about what the true dosage is, because both dosage 1 and dosage 2 have some probability of generating that outcome. As you get to 40 reads, there's much less overlap. The number of alternate reads where you have some uncertainty is a much smaller region. So one strategy for making this GT prediction, or what is the most, what's the genotype that has the highest probability, is to use the maximum likelihood approach. That just looks at the corresponding value of the x-axis and then looks up and says, okay, which of these distributions has a higher probability? So if I, for example, observe 12 reads of the alternate allele, that's this lower left example, then the dosage equals 1 has the highest probability. So my GT output from this algorithm would be the 0/0/0/1, so 1 copy of the alternate allele. If, on the other hand, I observed 18 out of the 40 reads to be alternate, my maximum likelihood prediction is 2 copies of the alternate allele. Okay, so that's maximum likelihood. In practice, the software you're using doesn't quite do that. It does something called maximum posterior probability, which means that it uses some prior information to multiply it essentially against the likelihood to come up with this new probability that accounts for the fact that not all of the possibilities are equally likely to begin with. So some of the information that goes into that may be, for example, things that are related to the alignment. So they have map quality scores. There are base quality scores from the sequencing machine itself. All that can get baked into this calculation. But the thing I want to focus on here is about exploiting information about your population. And that means if you're using a mapping population, using a prior that represents what is, given the parent dosages, you can know what the expected frequencies of each of those possible dosages are in your population. If you're in more of a breeding population or more of a random mating context, you can use a Hardy-Weinberg equilibrium and use that as your prior. In this 2019 paper by David and Felipe Ferrao, they came up with this more statistical approach called the proportional norm, which is a good default when you maybe don't fit any of these idealized situations. And that's actually the results I'm going to show you today are using that type of prior for the analysis. Okay, so in the classic way that VCF was developed, you have this kind of two-dimensional approach to characterizing your genotype. You have the prediction, and then you have a measure of the uncertainty. That's the GQ score. But in many cases, for quantitative genetic analyses, we can actually combine that into a single number, which is the expected mean or the posterior mean of the dosage, which can be fractional. It no longer is limited to being an integer number like the biological quantity it has to be, but these fractional values that can go in between somehow capture what is the uncertainty associated with the data that you have. And for a lot of the analyses that we want to do, in particular, anything related to additive values, whether that's in a GWAS or in a genomics prediction context, handle fractional values just fine because you're ultimately doing a regression of your phenotypes on the allele dosage, and they can be fractional values, and it causes no problems. And especially at lower depth, as I'm about to show you, there are clear advantages in terms of using the dosage instead of the genotype. So the two-letter code for the VCF file for the dosage is DS. And in this example that I had here, the posterior mean DS value is 2.8. So you can see the GT, which had the highest probability, was three copies of the alternate allele. That's what the 0.111 is. But there's some probability that even a dosage 2 could generate this pattern of 1 reference and 7 alternates. So that's why the value is 2.8. It's somewhere in between 2 and 3. As the total depth increases, eventually your DS approaches the GT because your uncertainty is getting less and less. So here's a simulation to show quantitatively what some of the benefit is of using, let's say, DS instead of GT if you're trying to make these predictions about genotype. And it's a population Hardy-Weinberg equilibrium. I'm simulating sequencing data, simulating read count data based on some real data I have from a targeted amplicon sequencing panel to figure out what's a reasonable assumption about the distribution of depth and whatnot. And the error you're seeing is the root mean squared between the true value in the simulation and then what the updog model predicts. And then I've got 3 colors here. Green is for diploids, up to red for hexaploids. The solid lines are the predicted genotype, the most probable genotype, and then the dashed line is this posterior mean. So what you want is lower error, and you can see across this whole range of data ploidy, and the x-axis is the depth associated with that particular variant for that individual. The DS dosage always has less error relative to the true value than the GT does. So you get some benefit, and that benefit increases as you get to higher ploidy and at lower depth, which is exactly what you'd expect. At low depth, you benefit from this using this expectation approach as opposed to simply picking the one value that's the most likely, because there could be another value which is almost as likely, and you don't have that, right? You can see for the diploid, you have to be at pretty low depth, like just a couple reads before, otherwise the difference between the GT and the DS is negligible, which is probably why you don't hear a lot about this outside of a polyploid community, because usually you got more than a couple reads, per sample. But you can see for tetraploids and hexaploids, the difference persists up to very high read depth, which is what many of you already know. And some of the typical rules of thumb we have about how many reads you need to have to achieve a reasonable error are really based on using the GT field. You can get by with a lot less depth and still have a very effective estimate of the dosage if you use the posterior mean. And that's something that Lindsay Clark has emphasized a lot in previous talks, and I don't think she's here today. Maybe she's listening online. But in terms of options, if you're very likely your service provider probably hasn't given you a VCF with a DS in it, again, because it's not something that a typical genomics center who works with diploids is maybe familiar with. But the Polyrad software can take an existing VCF and add the DS information to your samples. It's very fast. The main limitation seems to be it doesn't have a model for allelic bias. It doesn't seem to estimate also a lot of marker-specific quantities. So I like to use the updog package, which has a very good model, but it's definitely slower. You want to use, if you have big data sets, like, you can do 10,000 markers, a couple 10,000 markers in a day on a laptop computer. But if you're talking about hundreds of thousands of markers, you're going to need more significant resources to get that to run. The software I'm going to show you stores a lot of the information you get from updog, like the bias estimate parameter, the overdispersion. It stores it in the info section of the VCF file. So you have that there. You can go do filtering and other analyses after the fact to filter your data set. So this function is called just GBS, and it's in the polybreeder package, which you can find at this link on my GitHub site. It takes an input VCF file, it gives you back an output VCF file. You specify the ploidy, you have to give it the name of the prior you want to use, and this is the same names that are in the updog package, so you can check the reference manual and the tutorials for updog to know what those options are. There's no vignette yet on my GitHub site. The function is there and documented, but I'll be working to make a vignette there so you can see more clearly how to use it soon. Okay, so that's a little bit of more than a little bit of a digression, but I thought it was worthwhile to just talk a bit about that. Now that we're going to talk about the imputation methods. And as I alluded to at the outset, the first method I want to mention is this multivariate normal EM standing for expectation maximization. It's a classic method in statistics for imputing missing data. It has various assumptions associated with it, such as missing as random. But the idea is that we're going to treat the dosage vector for your population as a multivariate normal random variable. So the normal distribution, you all know, that's when you have a single number that is normally distributed. Now generalize that to a vector or more than one random variable, and that's what is called the multivariate normal distribution. And it has, instead of having a single variance parameter, it has a variance covariance matrix. It has all the variances plus the covariances of all those random variables. So the dimensionality is however big your population is. And the EM algorithm follows this procedure. In the maximization step, you impute the missing values by BLUP, conditioned on your estimate of the covariance matrix. And then in the E step, you then update your estimate of the covariance matrix based on what you just imputed, and you go until you converge. So I had developed this a long time ago when I was working on diploids. It's been available in the RR BLUP package in this function a.mat for the A matrix, because originally it was intended as an idea as a way of estimating the A matrix, or what we often call the G matrix, when you have missing data. But you can also still use it to impute the market data. One problem is that you get the values you get back lie outside the possible biological range. You will get values that could be less than 0 or greater than the ploidy, right, which are not possible. Statistics doesn't know that because that's not built into the theory. So the software, the AMAT software, just gives you the data back. The function I've written here in Polyploids called impute actually does truncate the data so that anything above your ploidy gets rounded down to the ploidy, anything below 0 gets rounded up to 0. So that's one difference between what the AMAT function does and this impute function. Otherwise, impute is more or less a convenient wrapper around this original function, and it rescales polyploid data to fit the diploid coding that's required for AMAT. And it also does things in the context of a VCF file. So you can give it input VCF, you can output VCF. You see here the possible methods that are available for this function impute. I've just described to you this EM algorithm. The population option is even simpler. It just replaces the missing value with the mean value for that marker, or if it's the GT, it's the mode, meaning the most common value. So that's an even simpler type of imputation, which works fine usually with SNP array data where you have a very low amount of missing data. But for GBS data where you typically have more, that's a little bit too simplistic. So then the third option for the method here is RF. That's the random forest. That's the next thing. It's again, classic machine learning algorithm based on decision trees. Here's a picture of a decision tree. This is the real decision tree that it generated for one of my simulated data sets. The way it works is that each marker is treated as the response variable in turn. And when you're looking at that one marker, then the software asks, well, how many flanking markers do you want to consider as the potential predictor variables? And you could specify that, that parameter. And then it builds this decision tree where each node of the tree, it's going to randomly, it's given a random subset of all the possible markers. And there's some reasons why that turns out to be a better approach. But then it looks to see, okay, which marker and what dosage value as a threshold should be used to best partition that such that you essentially minimize the variance of the training data set. So it's a splitting algorithm that continues to split and split and split, and it tries to split it to get the least variance around the mean for each of the two groups. And that does it that's one tree, and then it will repeat that many times and take the average prediction of all those trees. So the two parameters you have right now in the way I've written this software is you can specify how many markers to include on the flanking side and then how many trees you want to use. Surprisingly, even 100 trees seems to give good results. I've looked at some of the trace plots, and rarely have I seen examples where you need more than 200 trees before you don't really seem to get any much additional benefit. But that may be population size dependent. I'm not so sure about that. Okay, so here's a comparison of how well the EM and the RF algorithms do for imputation error in a tetraploid data set with 600 individuals. This is looking at one chromosome with 500 markers. Really, it's probably better to split your data set into separate VCF files by chromosome. Not essentially to do that, but that's a convenient way of parallelizing it because there's really there's not much information, or there shouldn't be much information from adjacent chromosomes when you're trying to impute on a different chromosome. anyways, two main things to take away from this. One, you can see that the blue dots are lower than the red dots. So random forest beats the EM algorithm, which is exactly what we saw when we looked at this in diploids 10 years ago. So that was not too surprising. You also see this strong dependency on minor allele frequency. The x-axis is the minor allele frequency. When you have a high minor allele frequency, there's more variance in the data, and so it's actually a harder prediction problem. If you have a very rare allele, you just impute the most common or the major allele, and you get right 95% of the time, right? So that's not hard. So that's why it gets harder to do that as the minor allele frequency increases. So for what I'm going to show you next, I've just only looked at markers with minor allele frequency greater than 0.3, which is it's kind of leveled out at that point. So it's a good way of taking a bunch of markers and distilling it down to even one number that I can show you some additional trends. So for example, what is the influence of population size on this imputation error? Here's a plot showing the performance of the EM versus the RF algorithm for populations of 200, 400, or 600 tetraploids. And what you can clearly see is that the RF algorithm improves as your population size increases, and the EM algorithm does not. So for small populations, if you only have a couple hundred, maybe it doesn't make much difference, but otherwise, I would recommend that you use the RF algorithm, even though it is more computationally intensive. I think it's worthwhile to figure out how to get it to work for your data set. Okay, so now that was population size. Now let's look at some other factors, such as ploidy. So here we've got 3 panels showing the error on the y-axis as a function of for ploidy of 2, 4, and 6. And then the x-axis is the minimum depth required for the genotype calling. So for example, if I have a value of depth threshold of 4, it means that I'm going to call as missing any sample by marker combination that has less than 4 reads, and I'm going to try and impute that particular instance. And what you see here is that, for one, the error goes up with ploidy. Okay, that's not too surprising. It's harder to impute data in a hexaploid than it is in a tetraploid and a diploid. The other thing you see is that as the depth threshold increases, you actually get higher error. So if I choose to, let's say, require 4 reads before I make a genotype call versus 6 reads, I'm actually better off in terms of the error using that lower threshold. And the reason I was a little bit surprised, by this at first, because I thought that, you have quite a bit of error associated with a genotype call with only 4 reads supporting it. So I was worried that using that in the training data set would lead to a worse prediction. But it seems that actually the trade-off, having the larger training data set, even though the quality of the data is lower, actually seems to win out in terms of these two competing effects. You've got the size of the training data set is larger as you lower the depth threshold, even though the error of that individual particular call is going up. So anyways, that's one thing that's a bit surprising, but the kind of take-home message here comes when we overlay this imputation error data on the original data I showed you in the very beginning about what's the error associated with the genotype call under the binomial model. So the green line is that data from the UpDog simulation that I showed at the very beginning, which has the opposite trend, right? With the under the binomial model, the higher the depth for that particular sample, the lower the error, and yet the error of the imputation is going up. So there's a kind of crossover that happens where the genotype call error curve intersects the imputation error curve, and that's what you see here. And that represents, if you will, what could be considered the optimal threshold for genotype calling in that particular data set. So it's hard to state that and think about it clearly. So let me try and break that down a bit more. Let's take an example. So in a tetraploid, for example, if I use a depth threshold of 2 under the, okay, you can see my cursor. So if I used if I would make genotype calls for all the situations that have only 2 reads, I would get an error that looks to be, I don't know, about 0.7 or so. That's the green line. But if I actually called it as missing and then imputed it, as long as I had 600 individuals, I could get a lower imputation error than I could if I used, so just throw away the 2 reads, essentially. I don't use them. and I just rely on the neighboring information from the other individuals in the population. And that crossover point tends to be higher for a hexaploid than it does for a diploid. Now, so I think it illustrates the potential that there are advantages to throwing away some data. Like in many cases, you might not want to make genotype calls with only 1 or 2 reads, but it depends, highly dependent on the population size and the structure. And I would be nervous about extrapolating the results you see here to any situation. I haven't done enough tests to know that. So I think that's something that's worth looking into in the future to see how robust is this kind of a recommendation to other situations. But it was an interesting result for me. And the parameter in the impute function that controls this is this min.dp parameter. So that's how you can control when you take an existing BCF, it can mask or not utilize the genotype calls for individuals depending upon what minimum depth threshold you want to use. All right, I think I'm out of time and I'm at the end, so thanks for listening.